PCR is a technique for copying a portion of a DNA strand outside a living cell. PCR uses DNA polymerases, which are enzymes used to replicate DNA. A primer is a short strand of DNA that's used to target a specific region of DNA and where that replication starts. This is a picture of a thermocycler that's used for PCR. DNA is found inside the nucleus of a cell. It can also be found in chromosomes. The target region for PCR ensures that any DNA sample is tested at that same exact location. Three major steps have to happen in order for PCR to occur. So, the three major steps are denaturing, annealing, and extension. Each one of these have a specific temperature range. For denaturing, it's at a very, very high temperature range of 94 to 96 degrees. The DNA strand is then cooled to a 45 to 65 degrees Celsius range. Then it's heated back up in the process of extension to 72 to 75 degrees Celsius. This is all done in a thermocycler.
So, let's say that we have a long, double-stranded DNA molecule, and we're interested in the highlighted part. These two strands would be the template strands. The first step of PCR is denaturation, meaning that we heat up our ingredients to exactly 96 degrees Celsius, almost as hot as boiling water. This breaks open all bonds between the two strands of DNA, so that they can separate from one another. The second step is called annealing. Here's where we need primers, and for our highlighted sequence, the primers would look like this. 3' end CGGATAC 5' end, and 5' end AGGTCAC 3' end. During the annealing step, we cool everything down to around 55 degrees Celsius, and this allows the primers to bind to their complementary sequences on the single-stranded DNA. The third step is called extension. We want to make the environment as optimal as possible for Taq polymerase to do its job. Okay, see, Taq polymerase is actually a really cool enzyme. We get it from a bacteria called Thermus aquaticus that grows in geothermal hot springs. So, not only can Taq polymerase withstand the heat during the denaturation step of PCR, but it also functions best at around 72 degrees Celsius. So, during extension, we heat everything back up to 72 degrees, and Taq polymerase latches onto the primers, grabs some free nucleotides, and assembles the new DNA strands. And that's pretty much all there is to it. A whole PCR cycle lasts roughly 10 minutes, and at the end, we've doubled the amount of DNA because each template strand of DNA is now double-stranded. So now we just continue cycling these three steps a couple dozen times. After every step, and the quantity of DNA gets doubled each cycle. 2 strands, then 4 strands, then 8, then 16, then 32, then 64, then 128, and so on and so forth. So, PCR actually multiplies DNA at an exponential rate. After around 6 hours and 40 cycles, we'll theoretically have 2 to the power of 40, or 1,099,511,627,776 copies. But that's easier to remember as just a whole lot of DNA that we can analyze further.
So the polymerase chain reaction makes a lot of DNA, and it contains the original genomic DNA. So this is all important because using those copies, we can run as many tests as necessary. One of those tests that we can run is an STR. What is an STR? It's a short tandem repeat, and they're places on the chromosome that contain short repeated segments of DNA. These strands are shorter than those of RFLP, so they're less susceptible to degradation. Not only that, but you only require a very, very small amount. About 1/1000th of a gram or less is required to run a test. The number of repeats is highly variable among individuals, so it's a very, very good test to be able to run. Now, the short tandem repeat that we're looking at is AATG. So we're looking at the DNA and wondering how many times does AATG repeat within a specific region. So we have 2 chromosomes here. Within this chromosome, AATG repeats 7 times. In this chromosome, it repeats 8 times. This is within the same individual. Well, you might be wondering, why 2 chromosomes? Well, we get one from mom and we get one from dad. So in this case, this individual would be heterozygous for this STR of AATG. If this individual was homozygous, they would have 7 repeats twice, or they could have 8 repeats twice. Another example, 4 repeat allele of TCAT. You can see within this region that it repeats 4 times. Within this region, it repeats 7 times. So this individual would be heterozygous for TCAT. We can see for this individual, they would have 5 repeats within their region of TCAT, and this would occur twice within their chromosomes. So they would be homozygous for a 5 repeat allele. If we were to look at each one of their chromosomes and we were to run their DNA, we could determine whether or not these individuals' DNA matches a crime scene. They would show up differently under an electrophoresis. The more STRs that are used, the smaller the percentage of the population that will have exactly the same STRs. This process is called multiplexing. The more markers that are used in a single analysis, the more likely that individual's DNA matches a DNA found at a crime scene. So the multiplexing takes more STRs and it runs more STRs within a single test. The FBI uses 13 specific STRs in order to determine a positive test. So 13 STRs is a very, very strong test. Now let's take an example. So what you're going to see is you're going to see this chart. This is an STR report. Now this STR report shows the frequency of these STRs within a population. Could you figure out which one of the STRs is the most common STR in a population? If you had said D5S818 is the most common one, then you're absolutely right. Whereas FGA is the least common out of the population. So if we took an example, what is the percentage that 2 individuals' DNA with 3 markers, TPOX, THO1, and D18S51 could match DNA found at a crime scene? We could look at the frequency that those 3 markers occur. So for TPOX, that occurs at 3.52%, THO1, 9.6%, and for D18S51, that's 4.3%. So we're only testing 3 STR markers. In order to do that, we need to put them in decimal form. So we're going to move the decimal twice over to the left so that 3.52% becomes 0.0352. We're going to multiply all 3 of the STR markers together, and we get an answer of 0.000145. Now, to answer the original question of what is the likelihood that two individuals' DNA could come up exactly the same, then you would just simply divide 1 divided by that number. So the likelihood is 1 out of 6,897 people. So, is that likely? Well, we can look at it in terms of a population size. What is the percentage that an individual's DNA could match someone else's DNA in a population of 100,000? If we were to take that same number of 0.000145 and we were to multiply it times 100,000, then roughly there's going to be 15 people that could match that DNA profile. That's only using 3 STRs, and the FBI uses all 13 of them.